Another method by which our cells can regulate and control the activity of enzymes and change the functionality of proteins is known as covalent modification. Now, generally speaking, in covalent modification, what happens is we actually transfer a functional group, a functional moiety, from one molecule onto that target enzyme, that target protein. So we actually covalently attach a group onto that enzyme, and that's precisely what changes the activity of that enzyme or the functionality of that protein. It can either turn on or in some cases turn off the activity of that enzyme. Now, there are many different types of covalent modifications that can take place inside our cells. So we can modify the proteins in many different ways. For instance, we have processes like methylation, acetylation, sulfonation, and many others that we're going to discuss in future lectures. But the one that we're going to focus on is known as phosphorylation. And phosphorylation is a very common method of covalent modification. It's a very common method by which we actually control the activity of enzymes and change the functionality of proteins. In fact, it's so common that nearly 30% of all the proteins, and this includes enzymes in eukaryotic cells, are actually phosphorylated. Now, why is this method so common? Well, as it turns out, phosphorylation is a very effective, very efficient, and a very convenient process. And we'll see why that is so in just a moment. First, let's actually discuss the family of enzymes that are responsible for catalyzing, controlling the rate of this phosphorylation process. So basically, just like any biological process that takes place inside our body is controlled by enzymes, this process of phosphorylation is also regulated by enzymes. And the family of enzymes that regulates this process, this family is known as the protein kinase family. In fact, inside our body, we have over 500 homologous protein kinases, which are responsible for regulating the rates of this process. So basically, these different protein kinases catalyze the phosphorylation of different substrate molecules, different proteins, and different enzymes. Now, what exactly does the process of phosphorylation actually entail? Well, in this process, that functional group that we transfer from some molecule onto that target enzyme or protein is the phosphoryl group. Now, the question is, what is the source of this phosphoryl group? What is that molecule that exists inside our cells that we can actually detach the phosphoryl group and attach it onto that protein? Well, inside our body, inside our cells, we have a very high abundance of high energy ATP molecules, adenosine triphosphates, which are produced by the mitochondria of the cell. And so because we have this abundance of high energy ATP molecules that contain the phosphoryl groups, these are the molecules that are the source of this phosphoryl group. So basically in this process, the protein kinase catalyzes the transfer of a terminal phosphoryl group shown in red from that ATP molecule onto a hydroxyl containing group, hydroxyl containing side chain found on that protein, the target protein. And because serine, threonine, and tyrosine all contain hydroxyl groups on the side chains of these amino acids, these are the three residues that are capable of actually using the hydroxyl group to accept that transfer, that phosphoryl group. So in this process, on the reactant side, we have an ATP molecule and the target enzyme or protein. And then on the product side, we basically have the ADP molecule, adenosine diphosphate. We have a single H+ ion that comes from this oxygen here, and we have this modified residue that is part of that target enzyme, the target protein. And so the red terminal phosphoryl group has been transferred onto this oxygen. Now, Because inside our cells we have this abundance of ATP molecules, it's inside the cells that the proteins basically undergo this process. And those proteins found outside the cells do not actually undergo this process of phosphorylation because outside the cells, ATP is not abundant. So reversible phosphorylation occurs inside the cells where the concentration of ATP is high and abundant. But proteins found outside the cells are not regulated in this method of phosphorylation because outside the cells we don't have an abundant supply of ATP like we have inside the cells. Now, the next topic that I'd like to discuss is, why is phosphorylation so common? So why is this one of the more common methods of covalent modifications? And as I mentioned earlier, it's because phosphorylation is very effective, very efficient, and very convenient. The question is, why? What makes this process so effective and so convenient? Well, there are 7 things that I've listed, 7 reasons that I've listed on the board. So let's quickly go through each one of these reasons. And let's begin with reason number 1. So in the process of phosphorylation, what we actually do is we transform a neutral residue to a residue, modified residue, that contains a negative charge. And because of the presence of the negative charge, it can basically break the old interactions and form better interactions, more stabilized interactions. And what that usually means is, if this residue is found inside the active site of the enzyme, the presence of this modified phosphoryl group, what it basically does is it forms new interactions with, let's say, the substrate molecule, and that can in turn change the activity, the rate at which the enzyme actually catalyzes that particular reaction. So number 1 is this process gives a net negative charge. And that's precisely what allows these new electric interactions to actually take place between this residue and that target substrate molecule. And this leads us directly into reason number 2. As a result of this modified negatively charged group found on this residue, because of this negative charge, that essentially gives this modified residue a high potential to form hydrogen bonds. And so the negatively charged oxygen atoms of the phosphorylated side chain residue can form hydrogen bonds with other molecules, for instance, the target enzyme or the target substrate molecule. And so this can increase the specificity of the interaction between the active site of the enzyme and that substrate molecule. Now, number 3 is the protein kinases are actually able to easily adjust the rates, the kinetics of this reaction. So depending on the physiological needs of the cells, this reaction can take place very, very quickly in a matter of seconds, or it can take place over a very long period of time. And the rate at which it actually, the rate at which it takes place and the rate at which the protein kinases catalyze the reactions basically depends on the conditions found inside our cells. If we have to undergo this process, the protein kinases can basically allow the process to take place very quickly. But if we don't want to carry out the process, these protein kinases can easily adjust the kinetics and slow down the speed at which this reaction actually takes place. Now, number 4 is actually what we spoke about previously. It's the fact that inside our cells, we have these high energy adenosine triphosphate molecules that can act as the source of that phosphoryl group. So we see that phosphorylation requires the transfer of this phosphoryl group from some molecule onto that residue of that enzyme. And these high energy ATP molecules, which are so abundant inside our cells, can easily be used and very effectively be used to actually transfer that phosphoryl molecule. Number 5 is, and we'll discuss this in much more detail, we have the process of amplification that takes place whenever protein kinases are involved. And that's because usually a single protein kinase enzyme can actually catalyze many enzymes at once. And so if each one of these enzymes itself carries out some type of reaction, the protein kinase, by activating all these other enzymes, basically amplifies the result. It amplifies all these different types of reactions, and that greatly speeds up the number of substrate molecules which are transformed to the product molecules. And we'll discuss this in much more detail in a future lecture. So activated protein kinases can be used to regulate many different enzymes and many different reaction pathways, and this can lead to an amplification effect in which we essentially amplify the amount of final product that we produce. Now, number 6 is whenever this reaction takes place, because essentially what we're doing is we're dephosphorylating this ATP molecule, and the breakdown of the ATP molecule is a very exergonic reaction. It releases energy. We see that this process is in fact thermodynamically stable, and the energy of the products is lower than the energy of the reactants. And in fact, about half of the energy, half of the free energy that is released in the dephosphorylation of this ATP molecule is stored inside this protein phosphoryl group complex. So dephosphorylation of ATP is a very exergonic reaction, and it releases a large amount of free energy. And that's precisely why this reaction itself is thermodynamically stable. In fact, because the products are so much more stable than the reactants, this reaction ultimately takes place in a single direction. So it takes place this way, and this reverse reaction doesn't actually take place, at least not at a very high rate. And that leads us directly into number 7. So if the process of phosphorylation is to actually be a convenient process, we have to actually be able to reverse this process, because if phosphorylation, let's say, turns on a protein's activity, we have to be able to basically turn off the activity of that enzyme by, for instance, removing that phosphoryl group. But as I mentioned just a moment ago, this process, as shown here, because this is so much more thermodynamically stable than the reactants, this process is essentially a one-way reaction. So the question is, how can we actually reverse this process? Well, instead of using the protein kinase to actually reverse the reaction, we use a different enzyme known as protein phosphatases. So inside our body, it's these protein phosphatases which are actually used to reverse the effects of the protein kinases. So protein kinases are responsible for phosphorylating that enzyme or the protein, but the protein phosphatases use a completely different reaction mechanism, a completely different reaction pathway to actually reverse the effects of the protein kinase and remove this phosphoryl group. And that can basically either activate or deactivate the activity of that enzyme. So if phosphorylation activates that enzyme, then dephosphorylation will deactivate the activity of that enzyme. And protein kinases basically phosphorylate, while protein phosphatases dephosphorylate. And they use the following hydrolysis reaction. So basically this is that modified residue of that enzyme. And what protein phosphatases do is they use a water molecule to basically remove this group and reform this enzyme that now contains this original residue that we basically had right over there. So these are the 7 reasons for why phosphorylation is a very effective and a very convenient process. So again, it produces a residue that is modified and contains a net negative charge. And that's precisely what gives it a potential to form many hydrogen bonds, and that allows it to actually interact in a much more stabilizing fashion with that substrate molecule. Number 3 is these protein kinases can easily adjust the rate at which the reaction takes place, and the rate really depends on the physiological conditions found inside the cell. Number 4 is we have an abundance of these high energy ATP molecules that exist inside our cells, and so we can quickly and effectively use them to actually transfer those phosphoryl groups. Number 6 is, again, with the ATP molecule, because the breakdown, the dephosphorylation of the ATP molecule is in fact an exergonic process. It releases an ample amount of free energy. That free energy is used to basically drive this reaction. And so the thermodynamic stability of the products will shift the reaction all the way to the product side. And so these products are much lower in energy than these reactants. Now, let's go back to number 5. Whenever dealing with protein kinases, typically a single protein kinase usually catalyzes many individual enzymes. And so instead of catalyzing one reaction, catalyzes many reactions at once, and so